Hey everyone, this video is all about triacylglycerol. So without wasting time, let's start. Triacylglycerols, also known as triglycerides, are the main storage form of fat in living organisms. They are insoluble in water, and because of their non-polar character, they are popularly known as neutral fats. It consists of glycerol backbone, a 3-carbon alcohol attached to 3 fatty acids by ester bonds. Fatty acids are the key components that determine the properties of triacylglycerol. They can be saturated, means no double bonds, or unsaturated, means it contains double bonds. Triacylglycerols are of two types, simple triacylglycerol and mixed triacylglycerol, that we will see later. Functions of triacylglycerols. First is energy storage. Triacylglycerols are non-polar and so they are stored in anhydrous form, an efficient way to store energy in the body. They pack more energy, 9 kilocalorie per gram, compared to carbohydrates which only produce 4 kilocalorie per gram. Second important function is insulation. Fat deposits containing triacylglycerol help insulate the body, maintaining core temperature. Examples include butter, coconut oil, olive oil, etc. A glycerol can be esterified with 1, 2 or 3 molecules of fatty acids forming mono, di and triacylglycerols respectively. Now we will see types of triacylglycerols. They are of two types, simple and mixed triacylglycerols. The key difference between simple and mixed lies in the fatty acids attached. In simple triacylglycerols, all 3 fatty acids attached to the glycerol are identical. For example, tristearine, where all 3 fatty acids are stearic acid, tripalmitin, where all 3 fatty acids are palmitic acid. But simple triacylglycerol relatively rare in nature. In mixed triacylglycerol, the fatty acid attached on the first carbon is saturated. On second carbon of glycerol, the fatty acid is unsaturated, and on third carbon, fatty acid can be either type, saturated or unsaturated. Mixed triacylglycerols are most common type found in nature. Most fats and oils we consume are mixed triacylglycerols. For example, butter that contains variety of fatty acids, making it mixed triacylglycerol. Now we'll see properties of triacylglycerols. First is difference between oils and fats. Oils are liquid at 20 degrees Celsius and fats are solid at room temperature. Oils have more unsaturated fatty acids or short chain fatty acids in triacylglycerols, whereas fats have long chain fatty acids and medium chain fatty acids in triacylglycerols. Oils are plant origin and fats are animal origin. Next is hydrolysis of triacylglycerols. It's a reaction that breaks down the molecule into fatty acids and glycerol. This process occurs in the presence of water and can be catalyzed by enzyme, that is biological hydrolysis, or by acids or base, that is chemical hydrolysis. First we'll see biological hydrolysis. In the living organisms, hydrolysis of triacylglycerol is an essential process for obtaining energy from stored fats. It takes place in digestive system and adipose tissue. Lipases break down triacylglycerol molecules at the ester linkage, releasing fatty acids. The fatty acids are then absorbed into the bloodstream and transported to cell where they can be used for energy production. Hormone-sensitive lipase breaks down triacylglycerol in adipose tissue. But mind well, hormone-sensitive lipase cannot break fatty acid at second carbon of glycerol. Next is chemical hydrolysis. First, acid hydrolysis. Strong acids like sulfuric acid can be used to hydrolyze triacylglycerol. This process is efficient but can lead to degradation of fatty acids. Next is base hydrolysis. This process is also known as saponification that we will see later in this video. Next is saponification. Soap formation from lipids happens through a process called saponification. It's a hydrolysis reaction where lipid, typically a triacylglycerol from fat or oil, reacts with a base like sodium hydroxide to produce soap, the salt of fatty acid, and glycerol. Factors affecting soap formation. First is types of fat or oil. Different fats have varying fatty acid chain lengths and saturation levels. These characteristics influence the soap's hardness and lathering ability. For example, fats with shorter or more saturated fatty acid produce harder soaps. Second is strength of the base. Stronger base like sodium hydroxide promote a more complete reaction, leading to a higher soap yield. Third is temperature. Higher temperature can accelerate the saponification process. The leftover glycerol from soap making can be used in various industrial applications like cosmetics or pharmaceuticals. Next is hydrogenation of lipids. It's a chemical process that adds hydrogen atoms to the double bonds of unsaturated fatty acids in a lipid molecule. This process alters the physical and chemical properties of lipids. There are two types of hydrogenation: full hydrogenation and partial hydrogenation. Full hydrogenation converts all the unsaturated fatty acids in a lipid into saturated fatty acids. This results in solid fat at room temperature. In partial hydrogenation, it adds hydrogen to some but not all of the double bonds of lipids. This results in a fat that is still liquid at room temperature, but with a higher melting point than the original oil. Now, what is the use of lipid hydrogenation? First is to improve shelf life. Unsaturated fats are more prone to oxidation, which can lead to rancidity. Second is to create solid or semi-solid fat from liquid vegetable, for example, margarine. Third is to improve texture, for example, making of cookies. Now there are some health concerns of hydrogenation. Partial hydrogenation can also lead to the formation of trans fat. They have been linked to an increased risk of heart disease, stroke, and type 2 diabetes mellitus. Because of the health concern associated with trans fat, many food manufacturers have stopped using partial hydrogenated oils. There are alternative methods for achieving the same result as hydrogenation, such as interesterification, which does not produce trans fats. Next is rancidity. It is a term used to denote unpleasant odor, taste, and flavor in foods due to deterioration of neutral fats, either due to hydrolysis or oxidation. Rancidity occurs in fats and oils on exposure to air, moisture, light, or bacteria. Hydrolytic rancidity is due to partial hydrolysis of triacylglycerol molecules due to trace of hydrolytic enzymes present in naturally occurring fats and oils. And oxidative rancidity is the result of partial peroxidation of polyunsaturated fatty acids with resultant formation of epoxide, peroxides, NADOls, and free radicals which can damage cell membrane. Lipid peroxidation is a chain reaction. We will make dedicated video on lipid peroxidation. Antioxidants such as vitamin E, vitamin C, beta-carotene are often used as preservatives in fat-containing foods to delay the onset or slow the development of rancidity due to oxidation. Lipid peroxidation can also be controlled by using certain antioxidants as food additives such as propyl gallate, butylated hydroxyanisole, and butyrated hydroxytoluene. Now we'll see the test to check purity of fats and oils. First is saponification number. It defined as the number of milligrams of potassium hydroxide or sodium hydroxide required to saponify 1 gram of fat or oil. Saponification number is inversely proportional to the molecular weight of fat. Higher the saponification number, lower is the molecular weight of fat. It is high in fats containing short-chain fatty acids. Second is iodine number. It defined as the number of grams of iodine required to saturate or taken up by 100 grams of fat. It's an index of degree of unsaturation. Higher the iodine number of fat, higher is the degree of unsaturation. Third is acid number. Number of milligram of potassium hydroxide required to completely neutralize the free fatty acids present in 1 gram of fat or oil. It indicates the degree of rancidity of the given fat. Normally, refined oils should be free from any free fatty acids. And last is Triacylglycerol number. It defined as ml of 0.1 normal potassium hydroxide required to complete neutralize the soluble volatile fatty acid distilled from 5 gram of fat. RM number is used to test the purity of butter or ghee. So this is all about triacylglycerol. Hope you get the value out of it. If you do, then please like and share this video and also subscribe my channel. Thank you.